Good evening. Thanks for coming. So, as Dennis said, I've only been here a relatively short time, less than 2 years. And when I first came down here, I didn't really expect to be studying harmful algal blooms right away. It's something I've studied in the past. It's something I'm trained to do, and I enjoy doing it. But I was doing other kind of research when I got here. And I think during this talk, you'll kind of see a history of what got me into in just the last year trying to study this phenomenon in the lagoon. It's a pretty important topic, as I'm sure you all know about. Because I've been here a relatively short time, I have a lot of people to thank because you come here, you don't know anything, essentially. You can read papers about the area, but it doesn't really tell you that much. And a lot of people have local experience, and you need to talk to them. And I'm indebted to many, many people. This probably isn't even the entire list. And not only people, but some of the organizations around here, our water quality people, our water management districts have been great dealing with. And some of the funding agencies in this area are starting, we ask them for money to do this kind of research, and a lot of times they actually agree to do it. My hope is that this list just keeps on getting longer and longer. So I actually gave the keynote lecture about a month ago at the Indian River Lagoon Symposium. And the title of the symposium this year was The IRL and Estuary in Peril. And if you actually saw that talk, you're going to see some of the same slides, so please excuse me. But the IRL, we all know what it is. It's this very long lagoon, which stretches about 40% of the coastline of East Florida. And it's obviously a critical and really diverse ecological resource for us, but it's also a really important economic resource. And the 2016 report that came out showed that it had approximately an $8 billion annual economic value. So, this is really critical to us, and we do need to take care of it. Now, the reason it's an estuary in peril, and the symposium was named after that, is pretty simple. It's because of what we call eutrophication. And if you're not familiar with what that is, basically when you do a lot of development around an area and you change the land use practices, we all like to live by the ocean, so we have a lot of houses. We strip a lot of the native vegetation away, which is a change in land use and how nutrients might get sucked up by our local plants. Once you start doing that, you increase the amount of point and non-point source pollution that could potentially get into a system that you're surrounding, and that's pretty much what happens, and this happens all over the world, and we call that eutrophication. And what that leads to eventually is degraded water quality and a damaged environment. And when we add in the effect of climate change, and in this case, our local waters are getting warmer. We don't have as many freezing events anymore. The IRL is so long, it encompasses 2 different climate zones. It's temperate and subtropical. It's getting warm enough that we're seeing what they call tropicalization. It's turning into one type of tropical system. It's losing its temperate characteristics. And when you add all that together, what you can end up getting is an increase in harmful algal blooms. So during 2016, I'm sure everyone here knows this, the IRL was in the national spotlight, and for not the reasons we want. You could find, this is just a sampling of the many stories that were written both nationally and locally on the harmful algal bloom problems here. A matter of fact, I think some of you told me, you saw me on the 6 o'clock news tonight. I just gave yet another interview on this problem. It's a hot topic here. And the reason that this was in the news, we had 2 major harmful algal bloom events. We call them HABs, but they're harmful algal bloom events. And in the north, we had a very large bloom of this species called Oreombra lagunensis. And we call it a brown tide. Obviously, the water looks like coffee. It's very, very chocolate brown color. And in the south, we had a bloom of Microcystis organosa. This is a cyanobacteria. It's a toxic cyanobacteria. And obviously, we'd call this a green tide. It's very green. That's easy to understand. Now, this organism here actually is not toxic, but it still has very harmful effects. And I'll talk about both of these a little bit more as I go on. So what is a harmful algal bloom or a HAB? And quite simply, it's just a dangerous concentration of phytoplankton. And here's an example of one phytoplankton that I study. This is actually a harmful algal bloom organism called the Cachiro sanguinea. And you can see it swims around. This is a dinoflagellate. It's a flagellated algae. It can move around. And you can imagine if all these swim up to the surface, you can get those massive concentrations that you see. But we'll back it up a little bit further. You may not even know what a phytoplankton is. If you were here last year, I gave a long talk on this. But phytoplankton that cause these harmful algal blooms are simply microscopic plants, and we commonly call them algae. Now, the one thing that's interesting about these, you just saw, well, they're swimming plants. So they're not exactly perfect. You couldn't just describe them like plants. They're also like protists. They have characteristics of animals too, but fundamentally they are plants. They conduct photosynthesis. And this group of organisms also includes photosynthetic bacteria. So these are eukaryotic organisms. They have a membrane-bound nucleus, but there's a whole bunch of these harmful algae that are actually bacteria, cyanobacteria in particular. And if you look at the size of these, this is a scale bar of 50 microns. This is that organism I showed in the video. And 50 microns is less than the diameter of your hair. So they're really small. You can barely see these things. And this is a case, these little spheres you see in here, those are individual cells of a colony of things. So these are really small. These are about 3 to 4 microns in size. This is a colonial organism, so a whole bunch of the cells kind of stick together. And this is actually a shot of that organism, Microcystis, that bloomed in the St. Lucie Estuary. So that's what that looks like close up, and you can see that nice green color that it has. So when you get massive accumulations of these different types of algae, you get all these different type of what we call basically colored tides. We have red tides, green tides, brown tides, and we just name them because of the color, simply. And a lot of the original, they used to call harmful algal bloom red tides. That was the common name. And the reason for that was these were some of the common ones we used to see. These are dinoflagellate blooms. They have a kind of a reddish pigment to them, and when they come to the surface, the water looks blood red. But here we get these green tides and these brown tides, and it's just the pigment that's in the organism that makes the color of the water. Now, these are really large blooms, but there's one thing it would be good for you to understand, and that's you do not need to have one of these massive blooms that you can visually see in the water for there to be a harmful algal bloom. There's a lot of what we call cryptic HABs. And this simple diagram shows biomass, the amount of something on this scale, on this axis, versus the depth in the water. I just made this up. But you could have one organism that's like this. There's a real lot of it up here in the water column, and it kind of varies with depth. And this could be a harmless phytoplankton. It's good for the environment. Things eat it and feed on it. And then this line here, you could have a very small amount of something buried underneath all this other biomass. And this could be highly toxic and be a harmful algal bloom. And you would never know it's there. You would just see, oh, this is dominating it, and you would barely detect that organism is there. But that could be just as dangerous as one of those very large biomass blooms you see. So you can have perfectly blue, clean-looking water, and there could still be harmful algae at high enough densities to hurt you in it. So just don't think because the water looks clean that there's nothing dangerous in it. That's something that you should always keep in mind. So just to drive that home, when you see something like this, and this is a shot of the St. Lucie Estuary during the Microcystis bloom we had this summer, this is very obviously a lot of biomass. There's millions of cells per milliliter. Milliliter is a tiny volume of water. That's incredibly dense. But at the same time, you could have a bloom of this organism, which is Dinophysis. It is very toxic. It's just 100 cells per milliliter. And it could be very dangerous. And that's 1/100% of that other organism. And yet it still could be there, and it still could be dangerous, and you'd never know by looking at the water. So before I rail on phytoplankton and you say kill them all, we have to realize phytoplankton are not always bad. They're almost always good. But occasionally we have some bad ones. And the reason they're always good is because they're photosynthetic organisms. They take in carbon dioxide and water, and in the presence of sunlight and chloroplasts, they produce glucose, which is a building block of sugar, and release oxygen. And if you look at the size of our oceans, which are filled with phytoplankton, the phytoplankton are responsible for greater than 50% of the oxygen we breathe. and the primary production. They're the base of the world's food chain. Without them, we would not be here. Hardly any life on Earth would be here. So they're obviously really important. So how are these algal blooms actually harmful when they occur? And there's three primary mechanisms, and I'm going to go through all three of these, and I'm going to start with toxins, but they can also do mechanical damage, and these high biomass blooms like we saw here this past summer, I'll describe what they can do. So for the toxin thing, I mean, why do the algae produce toxin? This is a common question, and there's two primary reasons why. One is what we call alleliopathy, and this is essentially chemical warfare on their competitors. They produce some of these chemicals that if they're with another type of algae and that chemical gets into that other algae, and this is a micrograph actually showing that. This is a dinoflagellate that was exposed to another dinoflagellate's toxin. It actually, in this case, makes the cells swell up and just explode. It kills it. So it's a way of getting rid of your competitors. They also produce toxin as a predator defense. Phytoplankton are eaten by what we call zooplankton, little crustaceans in the ocean. And this is an example of one right here. It looks like a little lobster. It grabs phytoplankton and it eats them. But if it eats a phytoplankton that's actually toxic, it can actually suppress its feeding. They don't like the toxin. It can suppress their reproduction, so this organism won't grow that well, if at all. And it can also suppress their motility. They get very slow and sluggish, and now they're susceptible to being preyed on by larger animals like fish. So they're essentially using this to get rid of the predators on them so they can grow uncontrolled. There's nothing to keep them down. Now, there's not just one kind of toxin. These organisms, there's a lot of different organisms that are toxic. In the IRL, there's probably at least 40 different species of algae that can produce a number of different toxins, and there's many, many different toxins. So I'm going to go through most of the common ones that we're aware of and explain what they do. So the first thing I'm going to talk about is what we call paralytic shellfish poisoning. And you're going to notice in all these names, they always say some kind of shellfish poisoning. And that's the initial way that most people got when these poisonings happen. They get it through shellfish because shellfish is the easiest way. Shellfish filter the water. They eat algae, that's how they get their food, and they can get large concentrations of toxin in them. And then someone goes to eat a mussel or an oyster, and they get a large dose of toxin. But just because it's shellfish poisoning, fish have it too. So it's not just shellfish, it just happens to be the way these things are named. So one of the most common ones is called paralytic shellfish poisoning, or PSP. And this is created by a toxin called saxitoxins. And the two organisms in the IRL that produce this and occur fairly regularly are this over here called Pyrodinium bahamense. It's a dinoflagellate. It's also bioluminescent, so if you've been in the northern IRL in the summertime and you've seen the water glowing, it's almost guaranteed it's this organism. Just remember, it's also highly toxic when you're out there playing in it. And this other one is called Alexandrium monolatum. This is a chain-forming dinoflagellate as well. Each one of these is an individual cell. And I've seen this in the IRL this year down in the St. Lucie Estuary. So if you eat shellfish or fish that have eaten or get exposed to these toxins, you're going to start out at a low dose of it with nausea. The more you eat, you're going to start to get paralysis to the point that your muscles for your lungs aren't going to work and you're going to die. And this is a really potent neurotoxin. Now, I've heard, I don't know if it's true, I think it's on Wikipedia, so take it for what it's worth, that, I actually know it's true. But, during the U-2 spy plane program with Gary Powers, and during the Cold War, and they were going over Russia, they actually gave the pilots suicide pills that were filled with saxitoxins. So it's a really strong neurotoxin. It's also classified as a chemical warfare agent. It actually can be weaponized, so it's really dangerous, and it's really common in water. The second type of poisoning you can get is neurotoxic shellfish poisoning, or NSP, and this is created by brevetoxins. There's multiple organisms that can make these toxins. The two that are common down here, most of you have probably heard of this in some form. This is called Karenia brevis. Really, really common on the West Coast. They have problems with this almost every single summertime, so much so that they've named it the Florida red tide. It happens a lot. It does bloom over here occasionally. The last time we had a major bloom of it was in 2007. There's other species that make it. This is Chatanella. This actually, we had a bloom of this right out in the channel here this summer as well. So if you eat something that has brevetoxin in it, you're again going to have nausea. You're probably going to vomit. You get numbness. A lot of times if you eat shellfish, if your lips tingle, stop eating right away. That's for all these toxins, that's a big hint that you're being poisoned. The biggest thing is respiratory distress. When they have these big blooms on the West Coast, people go down to the ocean and they start coughing, and they don't feel good. It gets into your lungs, and you will not be happy. The good thing about brevetoxins is I don't think they can really kill you. I don't know if there's a reported incident of someone dying from brevetoxins. I know they can hurt other marine mammals and possibly lead to death, but I'm not aware of a human dying of brevetoxin poisoning. It's still not fun if you get it. Another type of poisoning is diarrhetic shellfish poisoning. You've probably started to realize that the name tells you what's going to happen to you. This is DSP. This is created by a toxin called okadaic acid. The two organisms that can produce this that are local to IRL waters, this is Dinophysis. This is a species of Protocentrum. Now, these occur in the IRL, but the thing that is important to remember for some of these, not all, some of them occur here, but no one has seen them express their toxicity. And when I say that, the organism can occur, but yet not make toxin. But then something can happen in the environment to actually make it produce toxin and become dangerous. So just because it occurs here, it doesn't mean this toxin is always going to be there. But it always has the potential to be there. And again, if you get DSP, you're going to have nausea, you're going to vomit, and you're going to have intense diarrhea. It's not going to be a fun day for you. This is a really bad form of shellfish poisoning, and this is actually, unfortunately, pretty common too, like saxitoxins. This is called Amnesic Shellfish Poisoning, or ASP. And unlike all these other ones I've just showed you, which are created by dinoflagellates, things that I study a lot, this is actually created by a diatom, another type of phytoplankton. It's not modal. And here's an example of it here. This domoic acid that it produces is really nasty. And this is a fairly large problem on the west coast of the US, up near Oregon and Washington. But right now, where I came from, Rhode Island, our shellfish fishery is shut down up there. They're having a massive bloom in this right now. And they found very, very high levels in the local shellfish. The shell fishery is closed. If you eat something contaminated with domoic acid, you're going to have memory loss, potentially permanent brain damage, and death. This is a really, really nasty poison. This is one that most of you are probably familiar with. You're down here in the tropics. This is more of a, this is an organism that tends to live in very warm water. So down in the Caribbean, ciguatera, fish poisoning. Most people have heard of it in some form. It's produced by, again, this is a dinoflagellate. It makes ciguatoxins, thus the name ciguatera. And this is an epiphytic dinoflagellate. It actually grows on like seagrass and on substrates. So when fish come up and bite coral or they bite seagrass to feed or whatever they're doing, they actually ingest this and then they get the ciguatera toxin inside of them. And ciguatera, as far as I know, you can't get rid of it by cooking. So even if you eat well-cooked fish, the toxin remains. Ciguatera is another one. Obviously, nausea and vomiting, diarrhea, muscle pain, ataxia. Ataxia is when you can't control your muscles. You just kind of lose control. And eventually, you can get hallucinations. Ciguatera is another one that is not, I believe, fatal to anyone. It's going to ruin your weekend, and you're going to feel really bad, but you're probably not going to die from it. That's, yeah, cold comfort. The last thing I'm going to talk about are what are called cyanotoxins. And this is a really large class of toxins created by cyanobacteria. And you're going to hear a lot about cyanobacteria. This is the organism that grew in the St. Lucie Estuary that came from Lake Okeechobee. They're one of the most common forms of algae out there. They're pretty much everywhere across the whole world. Any freshwater pond is going to have cyanobacteria in it. Everywhere you look in the ocean, you're going to find cyanobacteria. It's a really common phytoplankton. They produce a whole suite of toxins. This is just a few of them: microcystins, BMAA, lingviotoxins, and all these are class of toxins. Neurotoxins, liver toxins, cytotoxins, endotoxins. They pretty much make everything. They're really, You don't know what you're going to get. And these all exist. We have all of these in the Indian River Lagoon. This is the microcystis that grew down in the St. Lucie Estuary. This is actually called Lingbia. It's an epiphytic cyanobacteria that grows on seagrass. Sometimes you see it floating in mats, and it produces a whole class of toxins which aren't very nice. And these are pretty common here. So they don't have to be toxic to be dangerous. So that was our tour through the toxins, but there's a lot of other ways these blooms can actually hurt wildlife. This is a pretty common one out on the West Coast. This is actually a very close-up micrograph of a diatom frustule, or a cell wall. And if you look at these setae that come off this cell, you see these sharp barbs on here. This is a glass shell. It's silica. That's what this diatom actually makes for its cell wall. So these are essentially little knives. When these organisms get into the gills of salmon, which is really common out west, they cut their gills apart and they kill them. And there's been massive fish kills on the West Coast in the salmon fisheries from this organism when it blooms. So another thing that can happen, and this is right from the Mosquito Lagoon, this is our brown tide that happened last year, and actually that organism's still up there. It's just not at high densities right now. If you have a case like this, these two fish tanks were filled with the same exact water with a bunch of phytoplankton in it. But this one over here had some oysters and shellfish in it. Different kinds of shellfish are really good at filtering the water. That's how they get their food. They take the algae out of it, that's what they eat. And you can see how quickly they clean this up. But when you have one of these particular blooms, this algae produces this mucus on the outside of its cell. And when this mucus gets into clams, shellfish, oysters, mussels, whatever, that mucus irritates them. They don't like it in their feeding gills. And they'll essentially clam up and they won't feed anymore. And they'll open again, it'll try it again. If it's the same algae up there and you just have this huge bloom that constantly goes on, eventually the shellfish will starve. They just will refuse to feed and will die. So when you have a bloom like this, you can have massive die-off of shellfish. Even though technically it's not toxic, it's still going to kill them. Now we get to the third issue with these big blooms, and this is when we have these really high biomass blooms, these things that have just like occurred here last summer with the microcystis and the brown tide Oreolumbra. So when you have that, and you have seagrass all along the lagoon, and seagrass is really important for a lot of animals and fisheries and such, they're on the bottom, and they need the sunlight to penetrate through the water to get to the bottom so they can grow. They're plants. Well, those algae take the sun away. That light from the sun never reaches the bottom. It gets absorbed by the algae, and it never gets down, and all the seagrass beds will die. They have no light, they're not going to survive. During the super bloom in 2011, massive amounts of seagrass beds were lost in the lagoon for this very reason. It just shaded them all out. So I'm going to go back to my phytoplankton are not always bad slide. They're not always bad. Most of the time they're good. But at night, large amounts can really be bad without being toxic. And the reason for this is the following. Plants at nighttime, they're not doing photosynthesis. They do what we do. We eat, we breathe oxygen, we produce CO2 and water vapor. Plants do the exact same thing at night. They use their stored up glucose, they take in oxygen, release carbon dioxide, and water. Now the problem is, when you have a real large amount of phytoplankton, and at nighttime they all start using the oxygen, you get this. The fish can't breathe. We call it hypoxia or anoxia. They die. The phytoplankton will live. They're going to die. This is from the Indian River Lagoon almost exactly this time last year. And this was caused by that brown tide up in the north. Yeah, it's pretty devastating. However, this is not a unique thing. So these harmful algal blooms, this just didn't start in the last couple of years. If we go back and look through the literature of what's been done here, we have over 50 years of recorded harmful algal bloom events in the IRL. Not a new thing at all. And back in the old days, there was a lot of dinoflagellate blooms. And I'll tell you one thing I did with this. All these are the pictures of the algae that are listed here. And they're all relative to the size of this one. This obviously it's about 50 microns in size, like the diameter of your hair, it's microscopic. But all these are relative to that one. And as you go through this, you can see all these dinoflagellates bloom. Here's the Carenia brevis I was talking about in 2007. But when we get to the last decade, this is what we see blooming here now. Now, what do you see right away? They're a lot smaller. We're starting to see this change from what used to happen to these different kind of phytoplankton that reach these really high biomass. They're what we call picoplankton. They're really, really small. And this has led a lot of people to start to think we finally hit a tipping point. We've hit a state shift where the IRL is not like it used to be. And we're starting to get these really, really large, extreme events. So now I'm going to get into the point of the talk. I'm going to talk about how, in this last year, I've got into really studying harmful algal blooms here. So a few days before that large fish kill I just showed you that happened in March of 2016, we knew this big bloom was going on up there. I had heard from I knew some people at that point in the St. Johns River Waterways Management District, and they do some phytoplankton monitoring, and they told us, there's this really, really large bloom up there. And me being a phytoplankton biologist, I was very interested in that. Well, it turns out another faculty member here, his wife, hung out with some skydivers who happened to be going over the Sebastian Inlet, and they took some pictures. And they brought the pictures to me, and they said, look, has this bloom made it all the way down to the Sebastian Inlet? I mean, is it taking over like half of the lagoon? I thought, wow, that's really interesting. I need to go out and sample that. I need to go find out just for my own curiosity. So I had planned to go find out if this thing is just spreading over the entire lagoon. And then the fish kill happened. Just a couple days after we were talking about doing this in the lab, it happened. It's like, okay, now we definitely have to do it. So I immediately went up there. I think I was there the day after the fish kill got reported. And we did a really quick sampling plan. The fish kill happened up here in the northern part of the lagoon. We looked at 6 stations. They're all starred here. And in the first 3 stations, that's what's presented here from station 1, 2, and 3. Now, this is not a normal microscopic picture of algae. This is what we call an epifluorescent shot. We hit it with one kind of light, and it glows another kind of light. And the light that it gives off is kind of indicative of the pigments that it has in it. This is chlorophyll A, gives a red fluorescence. But if they have other pigments, you get a different color of light out of them. So it kind of tells you a little bit about what's there. In this case, it was all this Oreoumbra. And there were greater than 1 million cells per milliliter, because we can also count them when we do this. And there were a few interesting things I found when we were doing this. One is you can see this is all the same thing, but these are much smaller. Well, there's more of them, but these are much smaller than these, which could be that these are growing extremely fast over here, but they're not growing as fast when you get down to here. And this is where the bulk of the fish kill actually occurred, too. And the other thing that I wanted to note about this is that was a really extreme fish kill. And I don't actually think the water went anoxic, which means there was no oxygen. It was just low oxygen. Well, the same problem that the clams have when they are trying to filter this thing and they don't like it, that mucus can get into the fish's gills. And when those gills get coated with mucus, they can't dissolve oxygen through their gills as efficiently. So I would guess, and I haven't tested it, but it'd be a great project for someone to do, that this mucus played a role in this, that the mucus got into the fish, and what they could have normally probably withstood with low oxygen, they couldn't. It just was a contributing factor to just the number of fish that got killed. So it was another one of these mechanical reasons why the fish kill was so bad. So the next thing I want to do is look down here and, in fact, answer my original question, had this spread? And in fact, it had not at all. There was a bloom down there, but it was a totally different group of organisms. And you can tell right away, remember I talked about the color? Well, you can see that this is not all red. They're all different sizes of things. Some of these are tiny. They're really hard to see. But you can see a ton of like green and yellowish fluorescence. That is indicative of cyanobacteria. There's a lot of cyanobacteria here. And other picoplankton, little tiny things we can't identify using a microscope. They're too small, and they have no identifying marks on them that you can say we know what it is. And in fact, I mean, it's kind of scary. We know almost nothing about these populations. We don't even know what species they are. But there's a lot of them out there. This was a very big bloom, almost as much as was up there, totally different group of organisms. So that was March. And in April, I had a second chance to get some samples. Now I was getting hooked. I wanted to know what was going on out there. And another professor here, Brian LaPointe, does this whole IRL sampling. If you can see all these little red dots that are all through this, he takes water samples for nutrients and other things at all these spaces. And I asked, can you bring us some water? We want to look at the phytoplankton that are in there, see if there's any harmful algae, or see if this bloom is in fact starting to spread or anything. It's been a month later. There were still being smaller fish kills. So he obviously agreed and started bringing us samples. And our first set came from up here in the Mosquito Lagoon. And in fact, even in April, that Oreo umbra bloom, that brown tide, was still going strong. There was a ton of it up there. And this is actually what it looks like through a microscope. If you were to come into my lab and look through a microscope, this is what you'd see. And they're just little dots. You can't really identify them, but it's pretty much all that's in there. Just a ton of these little cells. As we started to move down into the northern IRL and the top of the Banana River, again, it was totally dominated by this Oreo umbra. If you look at it, it looks just like the last slide, no different. It had taken over almost all this entire area. Now we only started to see a change, again, right where I saw it initially in March. It does not get down to the Sebastian Inlet area at all. It's pretty much still here in the bottom of the Banana River. But once we start getting into here, we start to see a shift. You can see a shift in that color, and you start to see other algae mixed into the water column. So we're starting to see a change. And once we get into the central IRL, it's fully different now. This is that cyanobacterial bloom right up here. There's a ton of it. It had actually gotten denser, more cyanobacteria. And then once we get down into the central IRL, we started to see what looks like a more normal phytoplankton population with a healthy zooplankton population with it as well. So that's what this is over here. They were feeding on it. It was starting to look a little bit healthier in some way, although this is a harmful algal diatom right here that makes domoic acid potentially. So there's still harmful algae around. And then when we finally get down to these, looking at these bottom samples down there, Jupiter and St. Lucie inlets, we saw a full-on, what you consider a healthy coastal-looking assemblage of organisms with zooplankton. And it's probably like this down there because there's so much flushing. There's 3 major inlets around this area, and it tends to get a lot of exchange with ocean water. So at this point, after seeing that data, I started to realize what a large problem we had on our hands. And I'm not the first one to think this, but how do we approach, if we want to know about harmful algal blooms and what we can do and understand them in the lagoon, how do we approach doing it? The lagoon is huge. It's really diverse and it's really complex. And scientifically, it's great, it's a challenge, but how do we deal with doing it? The way we doing it is to beg for money, because it takes a lot of money to do this kind of science. It's a lot of time. So that's exactly what I did. I got really energized and said, you know what, we need to do a better job. We just need to do this. I started calling every federal agency trying to get money. And I actually was successful. NOAA, the National Centers for Coastal Ocean and Science, they have a harmful algal bloom event response program. I called them and talked to them for many days to convince them to do it, but they eventually gave us money to actually start going up and monitoring the brown tide up there. They put it on their national website, and it was very nice of them to do it. They listened to us, and it's always good that the federal government gets interested in this. And the way we sold this to them, or the way I sold it to them, was that we can't go out there and continually take these samples and bring them back to the lab. It's just labor intensive. It's not the smart way to do it. But we have satellites in remote sensing that can look at the lagoon all at once. And if we can figure out a way to discriminate the bloom, we can look at its variability, its extent and development really quickly. You know, we can tell, well, is this that algae and is this a different kind of algae here? And the way we do that, discriminate what things are, is by looking at combining remote sensing with in situ optics so that we know the characteristics of the water, the optics of the water, that we can apply to what the satellites see or drones see that we fly over the lagoon to quickly assess what's there without having to go out there constantly and take samples. So in May of 2016, we've now moved on. We finally get the money in place. And in fact, my lab went out and did this. We took a bunch of transects up through the entire bloom area, all the way down to near the Sebastian Inlet. This is some folks from my lab. This is Aditya Nayak, Malcolm McFarlane, and Nicole Stockley. And we brought all our high-tech optical instrumentation out there and did transects through the water. We had a flow cytometer. We were taking water samples for Brian to look at nutrients. We did pretty much everything we could do. We generated a lot of great data. I will not go through it all. We looked at how the different organisms changed over space and time, essentially. We looked at how the dominance changed. We looked at the optics of it. This is reflectance. That's the actual color of the light that's coming off the water. The one thing I will know is that we were taking stable nitrogen isotope samples. And what stable nitrogen isotopes can tell you is if they're enriched or depleted in certain amounts, it tells you the source of nitrogen. This is essentially pollution in this case, that the algae are growing on. And in this case, the nitrogen was enhanced 3 to 9% for N15, which is fingerprinting for sewage. So it would tell you that the nitrogen that is making this very large bloom go is probably some kind of septic or sewage runoff. That's not a great surprise, but it was good to know. So that was May. We thought everything was going great. We're starting to get a handle on this. And then June and July came. This is a picture of Lake Okeechobee from space. You see all this green stuff? This is that cyanobacteria, Microcystis. And from space, you start to get a sense of the level of biomass of these blooms. We heard rumor, again, through the grapevine, you hear from monitoring that a very large bloom was developing in Lake Okeechobee. We, in fact, went to the satellites, this is Landsat, and looked at it, and sure enough, when we can see it from space very clearly like this, we know it's an incredible bloom. Well, the problem is we also had a lot of rain last spring, and when the lake gets really full, they got to release it. Canal 44 is here, and Canal 44 leads to the St. Lucie Estuary. So all this went there. This algae doesn't grow in the St. Lucie Estuary natively. It's a freshwater algae, and the St. Lucie Estuary is a salt estuary. But it can persist. It actually, if you put it there, it can actually survive for quite some time if the salinity stays low enough and actually grow. And this is highly toxic. Unlike the brown tide, this produces microcystin, which is a hepatotoxin. It'll wreck your liver. We also are pretty certain this produces some form of neurotoxin as well. So then, when that happened, we decided, oh, well, we got to go down there and start doing the same thing. So we went down and spun up the science machine and started to take measurements and do our work there. And at that point, I started getting involved with Martin County and started actually testing the toxin in the water, both during the bloom and after, to see, all right, at what point is the toxin low enough that the waters are safe again? So I got involved with them doing that. And then we also realized there's very little monitoring for algae down there at all. Now, there is some in the north. The St. John's Water Management District, about once a month, will take samples in about 6 to 8 locations. But no one's doing it in the south at all that I'm aware of. Now, Dennis Hanasak here has his Lobo array, the Indian River Lagoon Observatory, and they have to go out and do maintenance on them every 3 to 4 weeks. And while they're out there, they now collect water for us. So we can monitor at all the Lobo stations that are throughout this area. We can monitor for the phytoplankton that are there now. So at least we'll get some warning that something's going on. So we're still doing that today, and that should be a really nice data set to go with that environmental data that the Lobos collect. So I'm going to kind of summarize my summer of HABs in 2016. Up here in the north, we had the very large brown tide, the Oreo umbra. The pyrodinium, which produces the saxitoxin, bloomed again. It blooms almost every single year up in the northern part of the lagoon, and it bloomed again this year. We obviously had this very large cyanobacteria bloom that persisted around the Sebastian Inlet area. In Vero Beach News, there was a local bloom, only to pretty much restricted into the Vero area, where we started getting local people call us and tell us that they saw brown water everywhere around their docks. This was around the Johns Island area. So I actually got at, one of them gave me access to their dock. I went up and sampled it, and it was a stenoflagellate called Strepsiella. This is not toxic, but it was at high enough biomass that it could have caused one of these hypoxic events up there. So it was a nuisance bloom at a minimum. Right here in our canal, we saw a couple large blooms. We saw one of Chatanella, which definitely can produce toxins. It makes ichthyotoxins, fish toxins. We had some things we couldn't even identify, but massive blooms of them. We'd see them, you just walk out in the day and look out there and you see these really big blooms. Obviously, in Lake Okeechobee, we had the big Microcystis bloom, the blue-green algae bloom, which got over into the St. Lucie Estuary, and then because of the work we're doing with Dennis now, we found another harmful algal bloom. This one, we found it only at the deep water samples, the 3-meter samples of the Lobos. So they brought us back some water samples, and we found a real lot of this Alexandrium monolatum, which had been known to bloom in the IRL. This is one of the earliest reported harmful algal blooms here. And it produces saxitoxin, and there was a lot of it right there. Below, this. Now you've got to realize, we're not doing some massive monitoring program. These are just things we did on the side. If this is what we saw, you can imagine there were probably 2 to 3 times more things happening, more blooms that we just totally missed just for lack of having the ability to get out there and look. The reason that I'm pretty confident in saying that is if we look at just a tiny piece of the lagoon, this is what it looks like from space. And the first thing you see is, wow, it's really complex. There's little canals and there's little waterways everywhere. Some places are relatively undeveloped. Some places are highly developed. Some are going to have runoff problems. There might be uncontrolled septic systems there. And you're going to get good flushing maybe down the middle of the channel, but you're not going to get flushing there. And basically what that does is it creates what we call micro-niche or micro-habitats, so that you can get really specific conditions in one spot here, which aren't like conditions out there, and you can get a bloom, a local bloom of something bad or good in all these different areas. And then when you look at what the current monitoring is like out in the lagoon, and this would be what the St. John's River Water Management District does, they have up in this upper part, in the northern part of the lagoon, they do about, like I said, 6 to 8 stations spread out kind of across the whole lagoon. And what that really comes to is something like this. Once per month, and this would represent this, a larger area than this actually. If you take a sample here, do you think it's going to tell us what's going on here the entire time? The answer is no. There's no way. We just don't, we're not collecting enough. We don't know what's actually going on out there at any given time. So if you ask me what causes harmful algal blooms, and I tell you to go look at the research papers that have been written here, there's been about 12 of them in total, not a huge amount. But here are some of the reasons you will find that are published. What you understand from this is that this is not a simple problem. There is no way, if I just take one thing away, it's like, oh, I get rid of all the nitrogen pollution. All the HABs will go away. That's not the way it's going to work. These things have been going on for a really, really long time, and they're going to keep on going on. And there's not any one thing I could pick that's going to fix them. It doesn't work that way. It's much, much more complex than that. So obviously, this is an issue that we need to grapple with, and this is why it's so difficult to do this kind of science. I mean, there's no easy answer. I can't give you an easy answer. And that brings the second thing is, when we're trying to do this research, are we asking the right questions in our science? And I put this slide together with how much scientists tend to spend on each one of these. The bigger the text, the more time they tend to spend on it. There's 3 phases of every harmful algal bloom. It initiates, it starts, then it persists and it spreads, and then eventually it terminates, it dies. And when harmful algal blooms occur, everyone runs out there, oh my God, there's a bloom, and they take samples, and then they go look, and what caused it? I want to know what caused it. And everyone fixates on this, and I'm guilty of it too. But then what we don't do is then start staying out there and saying, well, how far, how long, why did it stay? What conditions allowed it to keep on going? And how far did it spread? Just like that brown tide I was talking about. Well, why doesn't it get down to the Sebastian Inlet? What's keeping it up in the north? Why doesn't it take over the whole lagoon? We can learn information from that. And then when it finally goes away, what made it go away? Was it temperature? What was it? This is how we start to understand mitigation. Is there something that is really critically bad for this thing that will wipe it out? We tend to fixate here. We don't fixate too much here. Now, the thing that's the most important, if we have to select what we're going to study, it's this real small thing down here. And I'll circle it and blow it up. Ultimately, the most important thing is the risk to our health. We are at risk. We need to know, above everything else, what toxins are out there, where they are, how much is out there, is it a dangerous concentration, and how trophic transfer means how is it getting into the food chain. We know it's in the algae, but is it getting into the clams? Is it getting into the fish you eat? Is it getting into the marine mammals? We need to know all this. So if we're going to pick one thing to study, this is the first thing we should study. We need to know this, because this is how we find out if we're in danger. And one of the most important things to realize is that, I showed you some of the toxins we know about, but there's a whole bunch of toxins that we call emerging toxins we don't know about that are out there. And if you start looking at these headlines, these get kind of scary. And these are recent things people are starting to realize, especially for cyanobacteria, because so many people are exposed to them, that there may be a link between dementia and ALS type symptoms and these neurotoxins that these things produce. And these, I just ripped these off easy headlines from, some of these are very valid journals. And you've got to remember, cyanobacteria are really common in the IRL. And yet we're not probably putting enough research into understanding this. And for the things we do understand, that we know these toxins are out there, we have tests for them, how are they getting into the shellfish, the fish, and the marine mammals? And we have proposed that we can use dolphins as like a sentinel species for human impacts. Because dolphins eat a lot of the same fish that we would eat, or we take from the lagoon. And in fact, we have started doing this. So, we can't sample live dolphins very easily. You can't go out and kill them. It's obviously against the law. But we do have dolphin strandings. And when dolphins die and they get stranded, they do necropsies on them and they save some of the tissue. And we can actually go back and look at those tissues and see if toxins were actually in them. And in fact, we've started to do it, and we have found both brevetoxin and saxitoxin in the local dolphin populations here. This research, that was a pilot study. We've now started a full study, and this is actually funded by the Protect Wild Dolphins Specialty License Plate. So when you drive around and see the nice little dolphin jumping over the sun there, this is the kind of thing that those license plates pay for. Some of this, the beginning of this kind of critical research. And not only are we looking at these toxins in dolphins, we're actually looking at these toxins now in their prey fish. And these are the fish that we can eat too. We share common things. And we're looking at it in shellfish as well, at least for some of these major toxins. And this is run through our foundation here at Harbor Branch. I also got funding from the Everglades Foundation. They're very interested in this as well. They funded us to look at these cyanobacterial toxins that are, originate in Lake Okeechobee and get all the way over here into the estuary and eventually even out to the ocean. So they funded us to actually start tracking the toxins all the way from Lake O, all the way out to the Atlantic Ocean. So that's a beginning. But how do we really improve and do this research to the best of our ability? The first thing we really need to do is to start with this remote sensing view of the lagoon and the waterways that go there. This is the only way we can get this world view, synoptic view of the lagoon and really target, if we see something of interest, to get out there and say, okay, there's something going on there, we need to go sample that. We can't just constantly sample it. We have to do it in a smart way. And we can use these kind of assets to start deciding where there's a problem and when we should go out there and do it. And these, we can do all of this. These satellites are up there right now and they're running. And we have drones that we can fly easily over the lagoon as well. In addition to that, we can do what we call coordinated high-resolution spatial-temporal mapping. Now, we already have, Dennis has his LOBO systems, the ORCA systems are out there, the St. John's River Water Management District has all these water quality monitoring stations already in place, which is a great asset for science. But we can supplement that. We can use these autonomous surface vehicles, which can take a whole bunch of instruments for nutrients, optics, pigments, they can take water samples, and actually map all in these little canals and areas that it's really difficult to get in with a boat. So these little vehicles can easily do this. They can also actually carry UAVs that can actually fly above, they can take off from it, fly above and take high resolution mapping while you're taking all these underwater samples, and you can match it up. And this is not science fiction. We have all this right now in place. We just need the money to do it. And we can get a much better idea of what's going on in the lagoon. Another thing we can do is bring advanced instrumentation. This is a holographic microscope that I developed in my lab. This actually allows you to look at the phytoplankton in the water in real time. It shows you everything that's in the water. You don't have to take a sample and bring it back to the lab. You actually get a live streaming picture of everything that's in there. And for some of these algae, you can pretty quickly identify what they are. And something like this can profile through the water, so you can find some of the ones that might be hidden down here. But you could also tow it behind a boat potentially and look at the distributions of algae over a large area instead of just taking these little samples here and there. Another big thing we can do, I told you, we know very little about these little tiny cells that grow in the water. We just can't identify them visually. What we can do, and it's a really a great emerging technique in science, is to use genomic techniques. So we can actually do what are called metagenomics and metatranscriptomics, and we can start to understand what species are actually out there and what kind of things they're actually starting to produce. We can look at how their RNA turns on and if they're making biosynthetic genes that are producing toxins. There's potentially ways of doing some better rapid toxin testing this way. But this is a critical thing we need to start doing because we fundamentally don't know what a lot of these organisms are. And as I've harped on already, you know, if you're not going to fund any of that other stuff, we really need to do this. We need to start doing this broad toxin screening and monitoring. We have to know what our liability is and our risks are to this stuff. And we fundamentally don't really know that well. So you're now asking, you know, how can we possibly do any of this? And again, it's hold up my sign, need money for science. We have to go to every agency that we can and ask them to support this science. We make a good case, they'll support it. I have personally written grants to every one of these agencies in the last 6 months, asking for research dollars to tackle this problem. The EPA, NOAA, they all have programs to do this. Even NASA. NASA has a very large program to look at harmful algal blooms. We've already gotten money from the foundation. We've got from the Everglades Foundation. We need to do more of this. We need the money to do it correctly. And the second thing is what's really important is we need better research coordination. There are a lot of people who are doing great research here. I mean, I've just started and I've met many people. But what we probably don't do enough of is really coordinate our efforts, talking to each other, sharing data, trying to, you know, don't duplicate effort, you know, and tackle the problem in a really good coordinated way. And two things that I'm personally spearheading now is I have written an FAU federal priority document to get FAU to try to get a HAB Center of Excellence here at Harbor Branch. But more importantly, I just put this grant in, and Gabby's out there, she helped, into this very large National Institute of Health and National Science Foundation program. And this is to establish a Center of Oceans and Human Health here at Harbor Branch. This is a 5 to 10 year program. They usually get refunded. We put in for $7 million to do this, which is a lot of money to us, but for the government, it's really not that much money. But if we get this funded, everything I just talked to you about, we can do, including, as part of it, we're going to do epidemiological studies on humans in this area to find out what toxins they actually have in them. So the CDC is going to work with us. We're going to try to do the entire thing I just outlined. And I'm really hopeful that we're going to get this. I think we have a very, very good chance to get it. And I will stop there.